Welcome back to the deep dive. We're here again, digging into complex ideas and trying to make them clear and useful for you, our listener, based on the sources you send our way.
Yep, that's the goal.
And today, wow, we are tackling one of the biggest questions out there. It sits right at the intersection of, well, chemistry, biology, philosophy.
The origin of life itself.
Exactly. How did we get from, just rocks and chemicals to life, to things that live, breathe, replicate? It's this huge, fascinating mystery.
It really is. And for this deep dive, we're looking at a really interesting article. It pulls together three thinkers, three really influential voices.
Right.
And what's cool is not that they agree, actually. They seem miles apart at first glance.
Yeah, that's what struck me.
But looking at their different takes, their contrasting views, it actually helps map out the whole problem space,
Okay, so who are these three thinkers? We've got James Tour. He's a synthetic organic chemist.
Very well known.
Michael Levin, a developmental biologist doing some really cutting-edge stuff. And Denis Noble, an emeritus physiologist. Could you give us just a quick snapshot of where each of them is coming from, just the basic idea?
Sure. So Tour is famously skeptical. He looks at the chemistry needed for life and basically says, hold on, this looks incredibly unlikely to happen randomly.
Okay, the skeptic.
Levin, he's focused on cells, but not just their chemistry. He talks about their cognition, how they seem to process information, almost like tiny minds.
Cellular cognition. Interesting.
And then Noble, he emphasizes the big picture, multi-level feedback loops, how the whole organism, the system, influences the parts, including the genes.
Right, the top-down view. So skepticism, cellular intelligence, and system feedback, they do sound pretty different.
They do. But here's the twist, the kind of aha moment we're aiming for in this deep dive.
Okay.
If you dig deeper past the apparent contradictions, you start seeing how they might actually complement each other. It's less like three totally separate views and more like maybe three chapters in the same story.
Ah, so a path emerges from early chemistry to complex life, linking these ideas.
That's the idea we're exploring, a continuous path.
Okay, I'm intrigued. Let's dive in. Where should we start? Maybe with the skeptic, James Tour. Lay out his case first.
Sounds good. So James Tour, Rice University, synthetic organic chemist. And his credentials, they're really impressive. Over 700 papers, hundreds of patents.
Yeah, he's done serious work, especially in nanomaterials. He's not just commenting from the sidelines.
Definitely not. And he uses that lab expertise, that hands-on knowledge to make his point. He often starts talks by showing these really complex chemical syntheses.
Right, the kind you do in a high-tech lab.
Exactly, using inert gases, super dry solvents, purification columns, all the stuff chemists use to get clean reactions.
Okay.
And then he hits the audience with that question: which of these steps took place on the Hadean Earth?
Oof. Yeah, that's powerful. Suddenly you're picturing this hot, messy, chaotic, early planet, not a controlled lab environment.
It's a very effective rhetorical move. It forces you to confront the sheer difficulty.
So what are the specific hurdles he raises? The big chemical roadblocks he sees?
He lays out 5 main ones. First is yield and purity. You run a reaction in the real world, especially under messy conditions. You don't get a nice pure product.
Right, you get mostly tar, maybe a tiny bit of what you wanted.
Exactly, a trace amount of some amino acid or nucleobase mixed in with all sorts of junk. Not nearly clean enough to reliably react in the next step.
Okay, hurdle 1, messy chemistry. Makes sense. What's number 2?
Number 2 is chirality, handedness.
Ah, the left-handed and right-handed molecule.
Precisely. Life uses specific versions, left-handed amino acids, right-handed sugars. But prebiotic chemistry, it makes equal amounts of both, a racemic mixture.
So, like trying to build something with screws where half have left threads and half have right threads, but you need only one type.
That's a great analogy. At every single step where handedness matters, your chance of getting the right combination is basically cut in half. Chain those steps together, and the probability just plummets.
Okay, that sounds, astronomically difficult for spontaneous assembly. What else does Tour point out?
Third, informational ordering. Life isn't just about having the right building blocks. They need to be arranged in a specific sequence to do anything.
Like letters forming a meaningful sentence versus just a random jumble?
Exactly. Randomly linking amino acids doesn't give you a functional protein. Randomly linking nucleotides doesn't give you genetic code. Where does that specific functional information come from?
Okay, so purity, handedness, information. What's the fourth?
Fourth is chemical integration. Think about a basic cell. You need lipids for the membrane, nucleotides for information, sugars for energy and structure, peptides for catalysis.
All different types of molecules.
And they all need to come together in the same place at the same time under conditions where they can all exist and react appropriately. Finding conditions that work for lipids and nucleotides and sugars, that's incredibly hard.
Yeah, one set of conditions might destroy another type of molecule.
Exactly. And then finally, number 5, temporal coordination. Even if you manage to make some of these useful molecules, they need to stick around long enough to find each other and react.
But early Earth wasn't exactly gentle, right? UV radiation, hydrolysis.
Right. Things fall apart quickly.
Yeah.
You need these delicate intermediates to survive long enough for the next step, another huge challenge.
Okay, so after outlining these 5 massive hurdles, what's Tour's bottom line? How does he see this resolving or not resolving?
Well, his conclusion is pretty provocative.
Yeah.
He basically says, looking at this complexity, it seems like only an intelligent chemist could make this work. Direct intervention.
Which, of course, leads to accusations of arguing for intelligent design or a god of the gaps. How does he respond to that?
He pushes back. He says the burden of proof isn't on the skeptic. It's on the scientists claiming abiogenesis happened naturally. He argues they haven't come close to showing chemically plausible pathways that overcome these hurdles.
So he's setting a very high bar for chemical realism, demanding more rigorous proof from the origin of life field.
That's his stance. A very chemistry-focused, bottom-up skepticism.
Okay. That's a powerful challenge. But now let's shift gears. Michael Levin. He comes at this from a completely different angle, right? More biology, less raw chemistry.
Totally different. He starts with things like regeneration. You mentioned the salamander limb earlier.
Yeah, why can a salamander regrow a perfect leg, but if I lose a finger, I just get a scar?
Exactly. Levin's work suggests the answer involves something called bioelectric pattern memory.
Bioelectric, like electricity in the body.
Yes. He's done these amazing experiments where he changes the natural electrical voltage patterns across tissues in, say, frog embryos.
Without changing the DNA.
Without changing the DNA at all, just
Right.
manipulating the bioelectricity. And he gets these embryos to develop extra eyes or limbs, sometimes in weird places.
Whoa, so the electricity is like a blueprint.
That's his conclusion. He thinks groups of cells use these electrical signals, these ion flows across their membranes, to communicate and build a collective map of the target shape, the morphology they're supposed to achieve. A cognitive map.
So cells aren't just blindly following DNA instructions. They're talking to each other electrically to figure out, okay, we need to build an arm here.
That's the radical idea. He uses concepts like morphological computation and attractor states.
Okay, unpack this a bit. Attractor states.
Think of it like a valley in a landscape. The cells are like marbles rolling around, and the final shape, the eye is the bottom of the valley, the attractor. The cells collectively compute how to get there by reading and changing the bioelectric landscape.
So the DNA provides the basic parts, the marbles, and the landscape rules, maybe.
Kind of. The genome provides the tools, the ion channels, the proteins. But the memory of the target shape, the goal, isn't solely in the DNA sequence. It's stored dynamically in these bioelectric circuits across the tissue.
Fascinating. So for Levin, where does life really begin? Is it about DNA replication or something else?
He pushes it earlier. He talks about a cognitive threshold.
Cognitive threshold.
Yeah. For him, the jump to life happens when some kind of bounded system, maybe even a simple vesicle, starts using energy gradients, not just to, stay stable.
But to actually predict and control its environment in some basic way.
Exactly. That ability to sense, predict, and act based on environmental cues, using energy flow, that's the dawn of cognition, and for Levin, maybe the dawn of life itself. It's more fundamental than just copying molecules.
Wow. That really shifts the focus from just chemistry to information processing and agency, even at a very simple level.
It's a completely different lens.
Okay, so we have Tour's chemical skepticism and Levin's bioelectric cognition. Now, where does Denis Noble fit into this picture with his multi-level feedback idea?
Noble adds another crucial layer, thinking about how different biological scales interact. His work on the Hodgkin-Huxley model of the squid giant axon is key here.
Right, a classic model in neuroscience. What did he find there?
He showed that even this very detailed molecular-level model didn't work unless you implicitly included control from the higher level of the neuron itself behaving as a system. It wasn't just ions moving. The neuron's overall state was constraining the ions.
So the whole is influencing the parts, even in that fundamental model.
Precisely. And he generalized this. He argues strongly that the genome isn't a sole blueprint. It's more like a set of tools or resources that the organism, the whole system, uses. His famous quote is, "The organism uses the genome, not the other way around."
That really flips the usual narrative, doesn't it? We usually think DNA dictates everything.
It does. And this leads him to ideas like downward causation and physiological time.
Downward causation, how does that work?
It means causation doesn't just flow up from molecules to cells to tissues to organism, it flows back down too. The needs or state of the whole organism, or even a tissue, can influence and constrain what happens at the molecular level, including which genes get expressed or which mutations stick.
Okay, give me an example.
Think about heartbeat regulation. The needs of the whole body for oxygen influence heart rate, which in turn changes signals down to the individual heart cells, altering their ion channel activity. That's downward causation. Evolution, Noble argues, selects for things at the organism level, like efficient running, and then fine-tunes the molecular parts to support that function.
So if a new behavior at a higher level works, evolution kind of locks in the molecular stuff needed to support it.
Exactly. It's a feedback loop across different timescales, milliseconds for cell signals, seconds or minutes for tissue responses, hours or days for the whole organism's physiology. He calls it the double clock.
So for Noble, the essence of life isn't about getting the molecules perfectly right from the start.
No, it's about functional coordination. It's about how the system organizes itself and maintains function across all these levels and timescales. That coordination is life.
Okay. 3 very different perspectives. Tour on the chemical impossibility, Levin on emergent cellular cognition via bioelectricity, Noble on top-down control and multi-level function. Let's try to compare them directly. Where do they see complexity actually starting?
Big difference there. For Tour, complexity is sky high right at the beginning.
Yeah.
You basically need something almost as complex as a modern cell from the get-go, chemically speaking.
No simple start.
Right. Levin sees it emerging earlier.
Yeah.
Once you have membranes trapping ions and some basic pumps, you can get these bioelectric circuits, maybe even in simple prebiotic vesicles. Complexity starts with that electrical signaling.
Okay. And Noble?
For Noble, complexity is relational. It emerges whenever a new level of organization appears that can constrain the levels below it. Each new layer of control ratchets up the complexity. It's not tied to one specific starting point.
And what about information? Where does that reside according to each view? Another key difference, I bet.
Absolutely. Tour insists information has to be chemically encoded from the start. The sequence of polymers, the chirality. It's got to be in the molecules.
Very bottom up.
Levin argues information emerges dynamically in the patterns of bioelectric activity. It's not necessarily tied to perfect chemical sequences, especially early on. It's in the voltage landscape.
More emergent. And Noble.
Noble sees information as distributed everywhere across anatomical structures, physiological processes, even interactions with the environment. It's not just in the genes or the voltages, it's in the entire functioning system.
Fascinating contrasts. Okay, one more comparison. Agency. The ability to act purposefully. How do they define that, or do they even talk about it?
Tour, coming from chemistry, basically sees no agency before you have high-fidelity replication. Chemistry is blind until then. No goals, just reactions.
Makes sense from his perspective.
Levin, however, sees agency appearing much earlier. Remember the cognitive threshold. As soon as the system uses energy to bias its future states towards goal-like attractors, like moving towards food or away from danger, that's the beginning of agency for him.
Even in very simple systems.
Yes. And Noble defines agency at the organism level. It's the organism's capacity to choose between different physiological options to stay alive and viable over time. Think homeostasis, adaptation. That's agency.
Okay, so Tour. No agency early on. Levin's agency emerges with basic predictive control. Noble agency is about organism-level choices for survival.
Wow, they really do seem incompatible on the surface.
They do. But this is where the source article gets really creative, proposing this gradient-first bridge to try and weave them together.
All right, tell me about this bridge. How does it work? Gradient first.
Okay, picture those hydrothermal vents again. Deep sea, volcanic activity.
Right, often cited in origin of life scenarios.
These vents naturally create chemical gradients. Think protons, like a tiny battery across the mineral pores, maybe iron sulfide structures. Okay, a natural energy source.
And we know simple fatty acids can form near these vents. These fatty acids can spontaneously form vesicles, little bubbles, protocells.
Like tiny bags.
And these vesicles could trap some of that natural proton gradient energy. Maybe pH differences inside and outside drive simple reactions. Maybe linking amino acids or nucleotides, albeit inefficiently.
So we're still facing Tour's hurdles, low yields, messy mixtures, wrong-handedness.
Yes, those problems don't just vanish.
Yeah.
But now, within this specific gradient environment, 2 new things might start happening, 2 emergent features.
Okay, what are they?
First, persistent energy harvesting.
Yeah.
Vesicles that are too leaky just fall apart. Vesicles that are too impermeable can't get resources. But vesicles that strike a balance that can maintain the gradient somewhat, they persist longer.
Ah, a kind of physical selection, even before genetics.
Exactly. Stability selection. Second, voltage modulation. Because ions are charged, maintaining that gradient across the vesicle membrane creates an electrical potential, a voltage. And if that voltage affects, say, how permeable the membrane is or how easily things get in or out, then the vesicle has a way to sense the gradient and act by changing its permeability.
Wait, that sounds like...
Levin's minimal cognition, right there.
Yeah.
A simple feedback loop between the environment, gradient, the internal state voltage, and an action, changing permeability. It's a tiny, primitive syntax cycle.
Okay, that's a really cool idea. Proto-cognition driven by a simple energy gradient, even without fancy proteins or DNA. But how do you get from these individual vesicles to something more complex, like tissues?
Well, vesicles can fuse. They can trap different molecules. Maybe simple peptides form that act as crude ion channels, improving gradient harvesting or control.
Slightly better pumps.
Right. Now imagine populations of these protocells. If they're close together, the ion flow around one can affect its neighbors. Their membrane potentials could become coupled.
So they start influencing each other electrically.
Potentially, yes, leading to collective behaviors. Maybe the whole group can sense and move along a gradient better than individuals, like a primitive biofilm coordinating.
Okay.
And crucially, when these vesicles divide or bud off new ones, the daughters don't just inherit the membrane lipids and trapped molecules. They could also inherit the electrophysiological state, the pattern of voltage across the membrane.
A bioelectric memory, passed down non-genetically.
Exactly. A heritable voltage pattern that biases the daughter vesicle's behavior.
And this is where Levin and Noble start to connect.
This is where it clicks together. You have Levin's idea of attractor states. The collective voltage pattern of the group becomes a target state, a goal.
Okay.
And then Noble's downward causation kicks in. That collective electrical state, that higher level goal, starts to exert selective pressure on the components inside the vesicles.
How?
If a random mutation happens in one of those simple peptide ion channels, and that mutation helps the whole group maintain its collective voltage pattern better or achieve its goal more effectively.
Then that mutation is more likely to be preserved and spread through the population.
Precisely. The higher level collective behavior shapes molecular evolution. The group's physiology directs the refinement of its molecular biology.
So it's like building a house floor by floor, where each completed floor helps determine how the next one is built. An iterative scaffolding.
That's a great way to put it. Selection is happening at multiple levels, acting as the glue.
Can you break down those levels of selection?
Sure. You start with chemical selection. That's Tour's domain, really. Only molecules and reactions compatible with the gradient environment persist. Basic physics and chemistry.
Right.
Then you get cognitive selection, which is Levin's contribution. Vesicles or groups that get better at sensing, predicting, and responding to the gradient environment outcompete the dumber ones.
Survival of the slightly smarter protocell.
Kind of, yeah. And then, as they aggregate, you get physiological selection, which is Noble's focus. Groove-level traits like how stable the whole film is or how well they buffer ions together become targets of selection. This drives the evolution of molecules that support group success.
Okay, this synthesis makes a lot of sense. Tor keeps it chemically grounded. Levin provides the engine for adaptation, this early cognition. And Noble explains how higher-level success locks in the lower-level changes.
Exactly. Tour's skepticism with the reality check. Levin's mechanism allows adaptation without perfect genes. Noble's downward causation provides the ratchet mechanism, making the process cumulative and directional.
It paints a much more continuous picture. So what does this integrated view mean for actually doing origin of life research? Does it suggest new experiments?
Absolutely. It points towards specific things we should be trying in the lab. For instance, trying to get polymerization linking monomers to happen inside these energy harvesting vesicles driven by the gradient itself, no external purification. That addresses Tour's realism.
Makes sense. Test the chemistry in the proposed environment. What about testing Levin's ideas?
We need to show that daughter vesicles actually inherit membrane potential patterns, and that these inherited patterns influence their subsequent growth or behavior, proving that this electrophenotype is heritable.
Okay. And for Noble's contribution?
That requires looking at populations. Can we design experiments where we select protocell communities based on a group-level trait, like the stability of their collective gradient, and then show that this selection changes the frequency of specific molecules or sequences within the vesicles, demonstrating downward causation.
So multi-scale experiments connecting group behavior back to molecular changes.
Exactly. It also means we need better ways to measure progress. It's not just about building a cell in a flask anymore.
Right. What kind of new metrics could we use?
We could develop things like a chemical plausibility index combining reaction yield with how common the starting materials likely were. That satisfies Tour's concerns. We could have a cognitive complexity score for protocells. How many distinct electrical states can they reliably achieve in response to signals? That captures Levin's idea.
Right.
And maybe a physiological integration coefficient measuring how strongly changing a group-level property affects the selection of specific molecules. That gets at Noble's multilevel feedback.
Using these kinds of metrics would give us a much richer picture of progress, wouldn't it? More nuanced than just pass-fail.
Definitely. And beyond the lab, this synthesis has, I think, some really interesting philosophical and educational takeaways.
Like what?
Well, it helps dissolve that old, tired debate between pure reductionism and vague emergence,
How so?
because it shows how they work together. Tor reminds us you can't ignore the chemical details, reductionism. Levin shows how new information layers, new rules can emerge from those details, crossing a threshold. And Noble shows how those emergent layers then loop back to constrain the lower levels, downward causation.
So life isn't just bottom-up or top-down, it's this constant loop.
It's a recursive construction project. Each new level builds on the last, but also changes the rules for the levels below.
That's a powerful image, and it forces different fields to talk to each other.
It has to. Chemists need to appreciate systems dynamics. Biologists need to respect thermodynamics and chemical constraints. Theoreticians need to engage with the messy reality of experiments.
And for anyone learning about this, the message isn't just, it's impossible or it just happened. It's more like,
Impossible might just mean we haven't found the right conditions, the right constraints, the right context yet. Look for how new rules emerge.
it really is a compelling synthesis. Tour is right. You can't just wave a magic wand over the chemistry.
No, the details matter.
But Levin might be right that cognition, in a basic sense, could start really early, driven by physics, not complex genetics.
Providing that crucial adaptability.
And Noble seems right that once you get higher-level organization, it takes on a life of its own and directs the evolution of the parts.
Creating that accelerating feedback loop.
So if we recap this braided narrative, it starts with geochemical gradients providing raw energy.
Powering proto-metabolisms.
Then ion-bearing membranes capture that energy and turn it into informational voltages.
Levin's minimal cognition.
These voltage-coupled collectives start working together, developing tissue-level goals or attractor states.
Emergent behavior.
And finally, downward causation locks in the genetic or biochemical tweaks that support those successful collective states.
Noble's ratchet, making it stick.
Each step builds on the last, addressing Tor's hurdles, using Levin's mechanism, and illustrating Noble's principles.
Yeah, it makes the origin of life seem less like a single impossible miracle and more like a series of plausible, albeit challenging, steps. The mystery shrinks, not because every detail is solved, but because we see a process how new constraints actually create new possibilities.
An iterative dance from rocks to reason.
Well put.
It leaves you wondering, doesn't it? What other huge scientific problems, things that seem impossible today, might just be waiting for us to connect the dots between different fields, to find those unexpected synergies?
What new constraints could unlock new possibilities elsewhere?
Something to think about. Join us next time for another deep dive.